Well, thank you very much. So now you have graduated and you're about to graduate, so you are now PhD students and we are increasing the tempo. On the other hand, if I am going too fast at any point, please interrupt the lecture at any point, right? There is, I have something like 50 slides for an hour and a half lecture. I don't have to go through all of it. It doesn't really matter. Again, it's better that something stays rather than I flood you with a lot of information which then can't be classified and it just evaporates. And I fully understand how it evaporates. It evaporates also if you, for 30 years in this business. So it's just a normal human nature, right? All right, so let me start with where we left up yesterday. So yesterday we somehow looked at this Hamiltonian and we somehow said, okay, I can find saddle point solutions and these in-principle shapes. And then we kind of said, okay, these shapes are really good if I want to, understand something like a shape of a red blood cell. And in the biological sense, I would say that pretty much this is where it stops, at least on a global scale, right? So now the membrane itself is still well described by this Hamiltonian, but there is a problem, right? I mean, it is a continuum system. And for example, any local interaction is not included. Even adhesion, which we kind of put in here, is basically a continuum force. So there is nothing such a notion of a protein in this. So we now have to find a way in which we can go and couple a protein to a membrane and maintain the particular, the fact that the protein is actually a particle, and then the interactions that this protein is actually inducing are local. They're not on the entire membrane, they're just on one place in the membrane, and they're not exactly on one place. That's going to be affecting the membrane in a certain range. The question is what range, how far, for how long, how much? These are all of the questions which we kind of have to somehow figure out and find a mathematical way to describe it. I mean, at the end of the day, this is now really a theory lecture on that problem. Okay, so, and with this we come to my chapter 4, and this is really to establish a model, and I will really talk about only one protein, and I will really look at that one protein in a membrane interacting with another receptor or a ligand or whatever you like with a counterpartner in the plane outside of the membrane. Okay, so I will look at only one bond problem, like single ligand receptor interaction. Membrane is, I have a protein in the membrane, this protein can diffuse, it is looking for its binding partner somewhere in the lower space, and they can attach and detach. So there'll be two questions which I'm trying to establish, and I'm establishing here as a model. One thing is what the protein does to the membrane, and the second question is what the membrane does to the protein. These are the two things which we have to model. We have to establish some framework to deal with these two questions, all right? And we will do it on the level of a single bond, all right? And not only in this part of the work, but this is really an array of students and postdocs which have worked on this problem or various aspects of this problem for nearly 15 years. So there is actually this is a fairly long story in terms of the work which has been done. So, and all of this work is also done in collaboration with the labs of Kaas and Gupta, who is actually graduated here. But now is at the CNRS in Marseille, Rudi Merkel in Jülich, and Mark Schwarzman at the Ben Gurion University in Israel. So now, why am I talking about this? Is because any basically cellular interaction or any way in which the cell learns about its environment and which organelles get to perform the function is by actually having proteins, and these proteins somehow interact on the membrane, and this is what I'm presenting here, an antigen-presenting cell which is interacting with these cells, but I could really show any other. This is a standard textbook picture, and there is a problem with this picture, right? It is really beautiful, right? It's certainly the picture of Mike Dustin, and I highly respect what Mike is doing. Mike is doing absolutely gorgeous work, but these pictures like this are somewhat misleading. They give you an impression that the membrane is just a plane. right? It's just a plane, and protein hangs on this plane, and membrane doesn't do anything except for the proteins to hang around, right? Well, that's not exactly true, right? And now this is from Mike's lab, a gorgeous picture of a neutrophil close to a surface and the undulations of this membrane. And I think Nir is going to give you 3 lectures actually as what actually drives these protrusions over here. There is much more to this. A lot of it is here driven by actin polymerization and depolymerization. But the summa summarum over here is that this membrane is really, really strongly fluctuating. And there are these proteins in these membranes which somehow have to survive these fluctuations. All right? And they have to work in this highly fluctuating environment. Now, let's have a look a little bit more quantitatively at these fluctuations. And here I'm just basically presenting results which we've done with Cornelia Monzel, which is now a professor in Düsseldorf. So what she built was something which she called a dynamic optical spectral something. But in essence, it's an optical trap. And you have a membrane, you take a membrane of the cell, you flood it with fluorescent dye. All right? And then what you measure, and then you can position this trap such that you are basically not in the center, putting the membrane not in the center of the trap, but putting it off-center in the linear regime. So that means that if the membrane goes up, the signal goes up. If the membrane goes down, the signal goes down. All right? Now, if I would be placing the membrane in the center, the membrane goes up, it's here, and I have no difference between the signal being here and the signal being there. So I can't recognize the fluctuation, right? So I have to shift myself off. I'm shifting myself off just enough that here the signal goes up, here the signal goes down, right? Because I have less and less membrane in that particular volume. And I can directly relate intensity, which I measure, to the actual height displacement of the membrane locally. Now, this is now a typical recording of the thingy, and what you can do with these types of setups is that there are 2 advantages. You can measure super fast, so you can really measure with, this is basically the fastest measurement that you can get, so this is the highest frequencies that you can achieve. And you can measure locally. You can, for example, measure in the center, in the rim, in the protrusion, in the filopodia, whatever you want, okay? It's a little bit like the technique which we see this morning, just using a different mechanism. So now, what is it that we measure? Now, I'm showing you first the fluctuation amplitudes, right? And now the fluctuation amplitudes here is only to show you that we are talking about very significant fluctuations. These fluctuations can be up to 0.5 micron large. All right? Now, 500 nanometers. How big is a protein? Orders of nanometer, let's say 10. Let's be generous, let's be 10. Let's talk about GPCRs, right? 10 nanometer. Okay, so this 10 nanometer object is being fluctuating in something with amplitudes of 500 nanometers. And now I often try to say, okay, well, this is like you trying to have a conversation with somebody who is in the lift and is being shooted to, let's say, 50th floor. So how can I have a conversation when, I'm here trying to, hi, I'm on the 50th floor, and then I'm brought down and up and down and up and down. How do I do that? All right, so this idea that the membrane is this flat, gentle, background is just simply not true. And you can just, with very little argument, see it. So what does it do? Why do we fluctuate so much? All right? Now, in the time domain, you can actually show that the cell really invests energy to perform this undulation. So this is not something that the cell has for free. And the question is, does the cell make these fluctuations because it wants to use them, or are these fluctuations a consequence of a lot of active processes which are happening in the cell? So the membrane happens to undulate a little bit more if it's active. Another thing which you can see over here is that this is now of power spectral density, right? Now, this is basically what is measured and shown over here by discrepancy of these two curves. One is measured in the linear response, one is measured in the fluctuations, and the fact that they are not the same, this is telling you that the fluctuation dissipation theorem has been violated, and that is actually the proof that they are active. Now, so now this is also a little bit puzzling, why would I invest energy in these fluctuations? This is a lot of energy that you have to put in. So what do I can I get something out? So this is another silent question which is going to be propagating through these conversations that we have today. Now let's measure them a little bit more precisely. Now what I'm showing you is the displacement spectrum, displacement probability of an activated and non-activated macrophage, okay? So what you see over here is this is a non-activated macrophage, the blue is an activated macrophage, and there are two interesting features, yes? This is the high probability distribution, so how much I have a displacement from my central position.
So this is greater than 1, I mean it's not normalized.
Well, they are normalized to the extent, they are normalized, this is like a Gaussian, right? But they're not Gaussians, and actually what you will see is that as a Gauss over here, they will be actually behaving as a Gaussian plus a Laplacian. So normal thermal fluctuations would just have a simple Gaussian. Now what we find in cells, and that's an interesting question, and that's why I ask you to have a look at your data, is that systematically when we measure in different cell types, these types of spectra comes out. Always we find a Gaussian and a Laplacian. Now, you could argue that this is not so bizarre because, the Gaussian fluctuations are always there. I mean, it's not so difficult to come up with a Gaussian process, active or passive. And, the typical standard process which gives you an active thing is an exponential decay. So I don't know anything about the origin of the active process or the Gaussian process or the Laplacian process. I have no idea where they come from. The only thing which I can state is that in all of the cells that we measured, we find a spectrum which is somehow a superposition of these two. All right? And obviously, if I measure in red blood cells, the origin is going to be completely different than in a macrophage. But from a point of view of a protein which is fluctuating, which is floating in this cell membrane, it doesn't care where the activity comes from. It just is. So, can I use this? Can I use this element of generality? Yes? Yes?
Typically, one would relate Gaussian fluctuations with something related to the central limit theorem. So here, are there correlations which are coming and violating the
So clearly we know that this is active. We clearly know that this is violating the fluctuation dissipation theorem. Unfortunately, I do not have a two-point correlation function measured. So my answer to your question is, if anybody has a two-point correlation function, please give it to me. I would love to have it. But no, we have a one-point correlation function, so with this we can only measure local property. But yeah, all right. But this is the fact of the matter. This is where we are. This is where the state of the art is at the moment. And there have been today couple of students showing data which could go beyond this limit. So if you're interested in how to push this, please let me know. Okay, anyhow, this is what you are. This is what you have. Again, I'm pointing out that these fluctuations are actually huge, right? So the excursion, which are going to this much larger length scales, and you can see that with activated cells, the high excursions are actually much more pronounced. Why? Why do I shoot a membrane, for 100 nanometers off? All right, now, let's look at what a protein does to a membrane. Now, this part is really done for the Gaussian membrane, because for the Gaussian membrane, you can do everything analytically, right? The nice thing about the Helmholtz Hamiltonian, it's a basically quadratic Hamiltonian, and if you model the bond as a quadratic spring, then you have a fully quadratic problem, and every quadratic problem you can pretty much solve on a spot. All right, so now let's look at the Gaussian case first, and I think this is always good. Later on, we can look a little bit more into non-Gaussian case. All right, so as I said, we have a spring, we have a membrane, and then what you can do, you can calculate the shape of this membrane, you can calculate the fluctuation amplitudes, and you can see this orange light thing at the back. So this orange line, this is the average shape by a protein which is pulling the membrane out its central position. This is the membrane at far distances from the bond, and this is about, let's say, 3 to 4 correlation lengths. This is how long it takes to saturate. And this is the fluctuation, the orange envelope. The light orange envelope is the fluctuation amplitude as a function of the distance from the central bond. So you can see that at the position of the bond, the fluctuations are suppressed, and as you're going far out, you again regain the fluctuation amplitude of the free membrane, right? As you would expect. I mean, no surprises here, okay? All right, now the interesting thing is that you can calculate the shape of this and really write it as a function of the correlation function or the Green's function. Basically, the Green's function is the correlation function of the free membrane. Now, that's the nice thing, okay? So that you can really write the shape problem and also the fluctuation amplitudes only as a function of the stiffness of the membrane, the excursion from the so the stiffness, the height of the membrane, and this is the height difference between unperturbed receptor and the membrane position, and this is the basically correlation function of the free membrane. And that's a nice thing, because you can all write in the Green's function comes out really nicely. So you have sources and you can play with this and then you can add it up and you can use all of the technology which exists for, let's say, Coulomb systems, right? And this one is vast. All right, so now let's look what the bond does in a time domain, right? I'm looking at the power spectral density now. In the power spectral density, what I'm plotting you here is the calculated and simulated spectrum as a function of the stiffness of the bond. Now, why is it important how stiff is the bond? Because if I am not stiff, if I'm stiff, I will really, if I'm completely stiff, just look at the limit, like if I'm rigid. At the point of attachment, I will completely suppress the fluctuations of the membrane. If I'm flexible, then the whole thing is going to wedge with the membrane, right? Up and down, right? All right, so as you are increasing the stiffness of the bond relative to the stiffness of a membrane, then you will see that in the high frequency regime over here, there nothing happens. This is the bending dominated regime, and you will see this power law of -5/3, as you would expect. What is really affected by the bonds is the low frequency regime. So over here, you see that the stiffer the bond, the stiffer the bond, the more there is an effect of the zero frequency limit. Okay, so now another thing which I want you to notice, that the zero frequency limit and this curve is not dependent on the extent of the deformation. So it doesn't matter whether I have pulled it a lot or a little bit, I will always have the same power spectral density. All right? This is what you can see here. This is if the membrane and the receptors are of the same size and they are not of the same size, the curve is actually the same. Okay? All right. A caveat here. Can we measure this? Can we measure this? Yes, you can measure this. Timo showed it really nicely. You have seen the slides, right? Now, you have to be very careful when you measure these things, because whatever you do, you never measure with infinite resolution. You always measure with a finite time and spatial resolution. Now, when you look at spatial correlation functions on the membrane, then these ones will not change the types of decay that they have with changing the resolution of the measurement. However, in the time domain, the Powell spectral densities actually change the Powell law of decays. So, when you're interpreting your Powell laws, please be very careful that you have actually taken the finite size effects into account. Okay? All right. Now, can we use this? I mean, this is a technique which we've been using in the labs now for maybe 10 years. It hasn't spread that much, but I think it's actually quite nice. So I'm going to show it to here. I added the slide this morning, also because I saw that some people are using RSCM. And then the point is that you can actually utilize the fluctuations to image the bonds. Right. So now, we have seen that you can measure the membrane in fluorescence, okay, but you can also measure in reflection, right? Now, when you measure in reflection, you're not in the height displacement in the position of the membrane. You're not limited by the optical resolution of the microscope. You're actually limited by the wavelength and the spectral properties of the system, right? So the resolution in the Ha, is of the order of 2 to 5 nanometers, where with lateral resolution is 100 nanometers. Okay? So now, if you just try to reconstruct the Ha field directly from the reflection interference contrast microscope, what you're going to do is a picture like this. This is a three-dimensional reconstruction. And what you see here was this is low, what is bright is what is high. Fine, right? Very nice. I have a kind of a blob of bonds here, a blob of bonds here, 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 and here. This is how you would interpret your data. Now, you can go and do a little bit more, right? You can, rather than you looking at the intensity, you can look at the variation of the intensity. So you're looking into a fluctuation map, which is over here. And because the fluctuation map is much more sensitive, you can actually see many, many more bonds and many, many more attachments than if you look in a direct signal. All right? Okay. So now, if I want to form a bond, I have to acknowledge that a ligand-receptor interaction is a stochastic process, right? So how do I model a stochastic process? We have seen this morning many times. You put in rates, K on rates for the forward process, K of rates for the backwards process. Mikhail is mentioning dynamics, and that's it. Yeah, well, that is a little bit difficult when you do it on the membranes. Why? Well, because the membrane doesn't let me to get close. Yeah? Hello, a K on, K off, who cares? Now, this was recognized long ago by Bell, Dembo, Hammer, Bongron. There is a class of people which have understood this. And they actually written these phenomenological rates. And I think pretty much anybody who has been doing biophysics for more than 5 years has used these rates, one way or another. I will tell you that these rates have actually never been calculated. They're all phenomenological, actually. And they say the following thing. I assume a detailed balance condition. This detailed balance condition tells me that my bond is a spring, right? And I will model my off rate by the force that this bond is experiencing. That's enough, because then if I have a detailed balance and then I have an off rate, I can calculate the on rate. And what you're going to see in the on rate, that the bond is actually, the binding rate is proportional to the energy that I can put into that bond. So it goes with H minus L naught squared. But mind you, I have an H here. This H, it's a nasty little beast. This is the position of the membrane. Instantaneous and local, R and T. Very annoying. Okay, now, we worked with these rates for a long time and, spend really a lot of time thinking, are these rates correct? And finally, honestly, just really now, last week, this paper got published where we actually showed that the first minimal model for the forwards and backwards rates on the Dembo system. Okay? And then you see that, all right, yes, you pretty much recover the same thing. There are little caveats over here. The caveat is that you have a contribution from the effective affinity. So when a bond is forming in solution, there is actually a binding enthalpy which you get. This binding enthalpy actually survives. So this is over here. And then there is a small entropic contribution because when you actually bind, you lose a degree of freedom. So I have two fluctuating ends. Now, when I'm inside, I make a bond. I actually lose a degree of freedom, and I have to pay for that. All right? And that comes in straight out over here. Now, there is a full derivation. In a full derivation, these rates look a little bit differently, right? They look a little bit more nasty. It looks some error functions are there. But then if you assume that you have a fast receptor conformational changes, a small binding site, so the way the ligand or receptor two proteins interact is small, following the size of the protein, and that the potentials of interactions have relatively strong curvatures, then you can actually get that. And this actually sets you the limits of applications of Denbo's rates. Okay? All right, so for the details, please look at this recent paper. And the difference is over here. Now, this bit over here comes again from the entropic argument in the off rate. As I say, please note and remember, the off rate goes linear in the displacement. The on rate goes quadratic in the displacement. Okay? That's the important thing that you need to try to put into your head. Okay, at least according to this model, right? So, all right, now let's go coarse-grained this. So what we have done this? All right, so in order to get to this result, what you do is you basically fix the position of the membrane and you integrate all of the degrees of freedom of the receptors. Right? So the receptor can fluctuate and it can access the membrane or can die. And in order to get to this result, you basically have to integrate this receptor degrees of freedom for the fixed position of the membrane. So basically, I still have a Ha here explicitly in the equation. Now, I can tell you, I can think about the system where, I can say, well, the membrane fluctuations are actually quite fast. These undulations, they happen, right? You have seen them, right? They go diggy, diggy, diggy. You saw the power spectral density. They're actually quite fast compared to the binding and unbinding of the receptor. Then what I can do, I can integrate the membrane degrees of freedom. So I can go and integrate given a probability distribution. Now, Now, I can assume that the probability distribution is Gaussian. Doesn't need to be. I don't care where this distribution comes from. It's something which comes from outside. So I will assume that this is my probability distribution. So what do I go and I say? I will calculate my coarse-grained rate as an integral of the probability distribution to observe a membrane at the position Ha times the off rate or times the on rate. Fair enough. I mean, this is just averaging. Huh? I've done this millions of times. Now, what are the consequences of such an averaging? Well, the consequences are very interesting. Is that I can now compare the rates of binding and unbinding for activated and non-activated system in average, and I will see that activation has actually increased the binding rate for orders of magnitude. Now, note, this is on a logarithmic scale. I go from something of the order of 10 to minus 5 to 10 to minus 2. This is a strong effect. This change in the spectra has produced this change in the on rate. It also changes the off rate, but much less. Now, look, this is a linear scale. All right? Why is that? It is quadratic in displacement. It is linear in displacement. When you integrate out, you get something which is on a power of high powers. So this reflects in this absolutely logarithmic dependence of the on rate on the position of the membrane fluctuations, on the average fluctuation amplitudes, roughly. Okay, what does this mean? I can look at now the binding affinity, K on over K off logarithm minus, off you go. So I will see that if I am a non-activated cell, I will be able to form stable bonds up to 200 nanometer away. But if I'm an activated bond, I will double my catching range. So now imagine this is for a macrophage. A macrophage which is activated suddenly can catch things which are much further because it's being activated. And I could argue, I can't have a proof, I can just argue that that's a very good investment. I put into the fluctuations, but I allow my efficiency to go up, let's say, a factor of 2, probably even more. Okay? So, and this is all what you get on a level of 1 bond. All right. Now, what can you do with this? Now this is now the same thing, but the result for the Gaussian fluctuations, because I also want you to know what there is also the same thing happens in the Gaussian fluctuations. It's the same dependence. It's not so strong because we don't have this exponential tail. Exponential tails contributes a lot, but even for the Gaussian fluctuations, we will find the same result. All right. The caveat: there is no such a thing as a protein binding affinity in membranes. That does not exist a priori. It is system dependent. Now, I can show you that I can vary the fluctuation amplitude of the cell or the membrane or the vesicle, whatever you want, or the height difference, and I can make a stable bond completely unstable. Right? This is what this model allows me to do. All right? So what does this binding affinity comprises? It comprises the standard binding affinity if I'm not coupled to the membranes, right? But this binding affinity is diminished by the price that I have to put into the deformation of the receptor and the membrane and in the entropy for making this bond. This is what comes out over here. All right? All right. Now, you can push these types of theory further. I will go through this slide very quickly because you can ask me later. You can now go and think, what happens if I now put a substrate which is a viscoelastic substrate? So I'm introducing an additional time scale in a system. And then I can play with this and I can say that, all right, if I work with something which is a highly viscous substrate, so very little deformations, then nothing particular happens. But if I will be breaking the bonds faster than I have a purely elastic substrate. But if I have a relatively weak, for this set of parameters, but let's say yielding substrate, so that I actually deform it, when I break this bond, it takes faster to actually rebind while the deformation is not completely lost in the substrate. So you will actually find many more adhesions or adhesive sites if in the optimal viscous range. Just from the effect of the rates, yes?
So only one pinning is considered here?
Here, one pinning is considered. This is the entire theory. I'm doing only one bond because you can't go and do many body if you do not understand one bond. All right? So let's just really stay with one bond. I will show you the many body aspects in a minute, right? So precisely, this is my last slide on the one bond case. And what I want you to remember is that, yes, you can integrate the receptor degrees of freedom, you get Denbo's rates. You integrate the membrane degrees of freedom, you get coarse-grained rates, which no longer depends on the exact position of the membrane, but they depend on the local fluctuation amplitude of the membrane, and they depend on the mean position of the membrane locally. All right? This is what I want you to remember. Now with this, let's say we have understood the one bond problem. Any questions? Now this is the time to ask, huh? No? Everybody's on board? Everybody's still, going on and thinking, oh my God, what is she doing? Oh, well, okay. Let's see. The many body problem. All right, so I want to look at now formation of many bonds. All right, one bond doesn't do that much in nature, right? You have to make many. But if I make many, then I have to take the fact that my membrane is an elastic fluctuating sheet seriously. Now, have you ever tried to hold hands with somebody on a boat when the sea is rocking? Well, it's much, much harder than when it is not, right? So it's basically the same story over here. The membrane is transmitting, although there is no particular interaction between this bond, and here I drew them of different lengths. They don't have to be of the same length. You can do the whole theory for different lengths. It doesn't matter. Then you will actually see that these two bonds will speak through the membrane, right? How does this happen? I'll give you an excellent example. Like I always, we always try to hang washing. Sometimes this happens that these lines are not perfectly tensed, right? So you put, your T-shirt over it, and your line does this, right? And now you try to put the next sheet over here, and then it's light. Then you somehow shake it, and then everything's flat again, right? It's precisely the same thing. It's exactly the same story. There is no difference between hanging washing on the line and making bonds in the membrane, except that now you're doing it in really, really high winds. So that's the story. So now you can go and calculate this. You can actually calculate explicitly. There is a formula for this, the interaction potential between two bonds which talk to each other exclusively through membrane-mediated forces. Again, have a look. I am putting this in a scale of kBTs. How much is an integrin bond in solution? Does anybody know? 10. How much is 10 kBTs? How much is a cadherin bond in solution? Maybe 4. How much is I don't know, name it. Typically, they go between 2 and 10. Why? Because you want to be able to control them with fluctuations. If you go with something which is 35 kBTs, like 100 kBTs, which would be a covalent bond, how much energy would you have to put in to break it? Endless. Too expensive, right? But your bond is strong, 5 to 10 kBTs, and interaction potential, which you get through the membrane, is of the same order of magnitude. It's not a corrective factor. It is a comparable factor, okay? So don't chuck it down the drain unless you really have to, okay? Often you have to because, you have to choose your questions, but if you want to think about adhesion, stability of bonds, then you should not be throwing it down drains. All right, okay. Now, there is another concept in biology which comes about, and it says, well, formation of a bond, a formed bond is a diffusion trap. And then you go and ask yourself, what the hell is a diffusion trap? Then you go and read a lot of biology books, and then you realize, I don't know what's a biology. Well, I don't actually understand. And the definition is, it's a region around the bond or protein and receptor where other receptors and other ligands are arrested. And that's a mechanism. In the physics group, in the physics departments, this cannot be a mechanism, all right? There has to be something more. So I will go and say, I can at least provide one mechanism for this. And I say, I am having a potential between this protein in the membrane and the pinning site. And if I allow for it, then I can actually calculate how much time does it take to come from the infinity or basically be in the close proximity and then leave. And I can calculate this for the first spin passage time. There's a standard tool in statistical physics. We know how to do that, right? And again, you will see that with small variations in surface tension and the small variations in whatever parameter that you choose that you have in a system, you can actually change this time from, being very small to super large. 10 orders of magnitude with very, very small variations in the system parameters. So here, I am changing the membrane compressibility, the membrane rigidity, surface tension, whatever you like. Small changes will do a lot because there is this strong sensitivity and long rangeness in the system. I told you it takes 2 to 3 correlation lengths to smooth this out. How much is that? What is the correlation length of a membrane? Come on, membrane people. Huh? It is at least 100 nanometers, right? So 100, 200, even to 400? Yeah?
Much less.
No. No. Go and do your calculation. Microns. Yeah, I'm talking about nanometers, 100, 200, 300 nanometers, half a micron. For a protein, this is enormous. Yeah? Coulomb interaction dies off after angstroms. This doesn't, hundreds and hundreds of nanometers, all right? This is huge for the protein, okay? And this is why this is so effective in actually arresting things around it, okay? Now that you have a model, you can go and say, well, I can do, many things. I can do long thermodynamics. I will take my Hamiltonian, my helpful Hamiltonian, I will propagate it in time. I need to come up with the Austin tensor for the membrane modes, which has been done in early 90s. I can plug this all in, and I can say my proteins. I'm not plotting all of the proteins diffuse on this membrane, and I say, well, this is an explicit random walk on the lattice. And basically, I'm only plotting, and then I can have another surface underneath, which is also not plotted. And the only thing I'm plotting is when the actual bond is established, because otherwise it will be just way too messy. You won't see anything in a movie, right? So you can propagate the system now completely. You have a Denbo's rate, where you can calculate the rate of interaction as a function of the local height, and you can have a full description of the membrane. Perfect, off we go. You can do many bonds, all right? At least numerically, lovely. Problem, yeah, this is 100 nanometers with 100 nanometers, and there are 50 proteins in the system. It's a bit annoying, all right? And in order to get the statistics, you have to do something like 300 and 400 simulations. So this is expensive. All right, this was maybe produced 10 years ago. Today you can push this, parallelize it, blah, blah, blah. Still quite expensive. And why? I don't need to simulate this membrane. I can do better, right? I can recognize my K on and K off rate. So rather than using K on and K off rates, which are height dependent, I can use my coarse-grained rate. All right? And I can say, well, for every site, for every protein, I will be having to look at my local environment. All right, so these are, let's say, a set of bonds, a little aggregate. I will look at my local environment. I will calculate what's the average height and average fluctuation amplitude at that position. And I will calculate my binding and unbinding rates. And I will propagate the system. I'm on a lattice, so I have a finite number of combinations if I look at only the first and the second neighbors. So I can even tabulate this, right? So, this can be really super fast. And if you do that, if you do this, then you can actually propagate the system for the kinetic Monte Carlo. Now, you do not need to do the membrane because it's integrated into the reaction rates. You don't need to do the Denbo's rates. You can look at your lookup table for your coarse-grained rates, and the diffusion you can still simulate explicitly of the protein. All right? Advantage? I can do 100 by 100 micron square meters, 5 microns, so cells or even 2 cells, and I can simulate this up to minutes or even hours, all right, on a normal computer, one single core. All right, now this code is fully available. It's highly parallelized if you want it to be parallelized, but you don't need to use it in a parallelized form. We can model up to 1 million of proteins. We can adjust the diffusivities of proteins. We can have different types of proteins with different diffusivities, different binding affinities. We can switch on and switch off lateral interactions if you want to. So the code is by now fairly versatile. And everybody would like to use it, please just let me know. All right. So the basic formulation of the code was published, some 7 years ago, all right? But there since have been some improvements over there. We can also simulate now things such as actual, we can also have spherical objects and not only linear objects. So this changes the fluctuation modes. You have to validate, right? Every theory that you come up, I have basically said, I'll throw out everything. I will only look at first and second neighbors, and I will throw out the membrane. Does this work? Well, I have my Langevin code to prove it does. I have to validate it against it, right? So there we are. I can look at the nucleation dynamics in the Monte Carlo and Langevin dynamics. They fit perfectly well. And I can look at the growth of various domains post-nucleation. And you can see the dashed lines are Langevin dynamics, and the dotted line, the full lines are the Monte Carlo code. So pretty much it's very stable for a very broad range of parameters. So it actually works quite nicely. All right, so that's the theory. We have a theory. Okay? I have nowhere shown that it works. So I have to prove that it works. And I have to prove that it works against experiments. Yes? I start with 0 bonds. No bonds. They choose whatever they want to do. So for the validation part, I don't pre-design anything. I randomly place them on the surface, and I let the system go. They cannot overlap, so there is an exclusion on a side-to-side, right? And that's it. And then they can have a look when there is a step where you diffuse. And then after the diffusion step, you have a look who's in your environment. Can I bind? If I can bind, then I'll try to bind. If I am bound, I will say, well, can I unbind? I'll try to unbind following a kinetic rate, Boltzmann weight. And that's it. Yes? Well, there is no memory in the time dynamics in the sense that only memory is encoded in their environment, right? So their environment has a finite time, right? So you have proteins in your environment. So if I have another one over here, I will not be able to move to that side because somebody is over there, and I will memorize this for a little while. All right? That's the first thing. And the second thing is if there is a bond, if there is an established bond next to me, my membrane fluctuations locally and my height is going to be different, so that's going to be recognized by the rates. Okay, so I am encoding the environment, but all of the encoding in the environment is shuffled into the rate. Okay, so in that sense, the rates account for this effect. And then in every step, I have to relook into my environment and choose my current rate. So the rate is a time-dependent issue. All right? So I had a lovely example over here. Over here. So for example, now, assume that you start with one step. You perform a Monte Carlo, and then these two bonds have unbound, right? I have lost them. Statistically, there is a probability that this is going to happen, which means that all of the other rates will actually have to change color. So here, I lost one over here, right? I lost these two, right? But that means that this bond now will unbind with much higher probability than before, right? Because it's now exposed. So, and this is what you have to update in every step. Okay? Yes?
So during pinning and unpinning, in that step where the membrane is fluctuating, so their excess movement can be added by exocytosis and also, the reverse of that.
Well, that bit is not in the code. If there is a way to parameterize this, that's easily changed because the only thing that you change in that respect is actually the tension of the membrane, right? And if you tell me, well, at time t, change the tension of the membrane for 10%, then I'll change the tension for 10%. No problems. But you need to tell me, I need to have a way to parameterize this process, right? And that has to come from somebody who is doing an experiment. I mean, I can imagine, okay, I can, turning off the tension gradually and then look. I mean, I can come up with millions of ways to do these simulations. It's really not that hard, but, why? What am I trying to model? Okay, I can explore this forever, but the reason is there anything I can show or prove or disprove with this model, all right? If there is, then I'm happy to engage. All right, so now let's see whether this works in experiments. And when I say now in experiments, I will exclusively look at the mimetic system. Why? We spoke about this in the last few lectures. In the mimetic system, I know exactly what I put in, and I know exactly what should come out. I have not fiddled with it. I have no active process to regulate the tension, to regulate the density, to regulate the activity, to regulate the fluctuations. It's dead. It is what it is. It's a physics system. So if I want to validate a theory, I should be able to, if I can't make it work with the experimental system, which is a purely physics system, then I have a serious problem with my theory, right? So that's why we'll be looking into vesicles. We will be looking basically into vesicles which are decorated with cadherins. Cadherin is a type of a binding protein, and these vesicles will be offered a substrate which is also decorated with cadherins. So the way cadherins recognize each other, this is one cadherin, this is another cadherin. These cadherins talk to each other by basically binding on their outer domains. All right? And this is known from biology. Now, there is a lot of more things that people know in biology, which I can't put in this model because it's too delicate. But, we can do that. Now, we have a cadherin here. This is actually a vesicle. It's closed, and then I have a surface. And these cadherins are actually floating in this, actually deposited in a supported bilayer, so they can actually move. All right, so they're really on the fluid membrane, which is below. Okay. Now let us calculate what we expect. What I first want to calculate, I want to calculate how much time is it going to take to make a stable nucleation domain. Something which I can see under the microscope. One bond I will not be able to see. Okay? So I need to think, okay, how long does it take to make a stable nuclei? All right, that's what I want to calculate. And what's going to be the size? And here you look at the capillary approximation, you balance the bending versus tension terms, and off you go. You come up with something. And that actually gives you the number of bonds. Because the size of the protein so basically tells you the number of bonds which you have to nucleate. Surprisingly, it's actually quite little. You need 2 to 3 bonds to nucleate. Okay? You can also calculate the nucleation time. For that, you need to write a master equation. We have seen these master equations over the last 2 days in many different systems. You just write it with appropriate rates on and off, and that's it. You solve that. Yes? Question? No? No? No. Okay. You can solve this analytically within certain approximations, and that the approximation is that the shape of the membrane as you're going from 1 bond to 2 bonds to 3 bonds is not changing drastically. That's the approximation. Under this approximation, you actually get an explicit formula for the nucleation time. And you can now plot this fluctuation time as a function of the height difference, and then also I'm plotting, okay, here on these sets of parameters, I will need an infinite amount of bonds to have a slow stable nucleation, so no adhesion over here, I will never have a stable adhesion. And then as I go down over here, then I see I here need only 1 bond to nucleate, and typically in this regime, and this is where we typically make most of the experiments, these are some points from the experiments, you need about 2 or 2 to 3, 4 bonds to nucleate. Okay? All right. Now again, have a look at the nucleation time. There are 9 orders of magnitude here. 9. I can't cheat that, right? If the theory creeps up, I mean, I can't go and say, well, this is a small effect. So I need to see something massively happen in the experiment. All right, so now Kea and Susanne Fentz did this experiment. It took a long time, and actually, to be honest, the way it worked, it was like this. They started working on the cadherin system. We worked on stronger systems before, right? So biotin-avidin, integrin systems, and that all kind of worked and was consistent. And the cadherin system, you would make an experiment, and it would just show something. And then when you make an experiment, it would show something completely different. And then you make it again, and it's something fifth. So 2 years into her PhD, Susanna took the cadherin project and threw it into the dustbin. And said, well, I need to graduate, let's go back to the biotin-avidin system. It works. I put the vesicle, there is a domain, it grows, I can measure its speed, off you go, everybody happy. This mess with cadherins, no. Now we then, actually, 4 years later, calculated this. And said, well, Susan, you remember this cadherin data? Have a look, okay? So then we worked a lot on actually establishing the codes and all left and right. And what you see over here, as you're increasing the fluctuation amplitude, we'd go with 1 or 2 nucleation sites to actually quite a few, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? If you are only decreasing height, again, you go to 1, 2, 3, 4, 5, 6, 7 nucleation sites. So this is from 1 to 10, roughly. And then you decrease both. You increase the fluctuations and decrease the height, and you basically go for something which you don't actually see the domain forming because the entire contact zone is slowly coming, and it's because the bonds are forming all over, because one bond is enough to nucleate. Yes? How does the
nucleation rate is increased?
You know, I told you yesterday. You change the osmotic conditions. When you change the osmotic conditions, you change the area to volume ratio, which is going to change the fluctuation amplitude. It's very clean, right? No cheating. Okay? And what I show you on this data, the actual, really, these are the actual fluctuation amplitudes, the actual height difference. This is really calculated for the parameters of the experiment. All right, so now we're kind of convinced that this works, that this theory has picked some things right. Okay, no fiddle. There is no fitting parameter. Because we can measure everything. We can measure the height, we know the size of the cadherins, we know the binding affinity of cadherins, we know the fluctuation amplitudes. That's all what we need. All right. So, okay, so we are happy. Now you can go a bit further and think about what, now this is the entire growth process, and now this is a slightly different system. This is a biotin-avidin system. And this is a comparison with some data which came from Jake Grove's lab. And where they observed that if the proteins on the surface are immobile, then you get kind of a flat adhesion. But if the proteins on the surface are mobile, then they get this kind of a ring-like structures inevitably. So we looked at this, and this is where we see, okay, in the mimetic system, you get absolutely the same. This is one of these ring structures. And this happens very simply because as the things are starting to bind, the proteins from the bilayer are actually diffusing in the contact zone. They get trapped close to the edges, and they basically clog the whole system. It's a dynamic trapping problem, and this is what we can really simulate it as a full-size system. Okay? And now you see the time over here, this is something like 60 seconds, right? So, and this is the number of formed bonds. We are talking about 100,000 bonds. Okay? So no, this is not partially fluid. This is a partially fluid system, and this is, sorry, this is wrong. Yeah, so this is partially fluid, so the diffusion is slow. So these ones come inside, and they get trapped before they can move in forwards, right? And over here, the system is fluid, so the proteins which are coming from the outside, they can come and reach the entire contact zone. That's how it happens, right?
How you made the vesicles with E-cadherin?
How do we make the vesicles with E-cadherins? Yeah. Okay, so this is really a more question for Susanne and Kea than for me. But in essence, what we do is, and what is typically done, is we do electroswelling, and then with electroswelling you add, so here cadherins are actually, you're looking only at the outer domain in a dimer form, and it is tagged by covalent bonding to SOPC. Sorry?
These are artificial lipids or cell derived?
There are artificial lipids. I presume they're artificial lipids, right? Don't know, honestly. But I presume they're artificial lipids. They are not cell-derived constructs. I mean, cadherin has a huge extracellular domain, intracellular domain, and transmembrane domain. And you chop it off at the extracellular side, and you tag it to the lipid. All right. Now, with this, we're kind of convinced, well, at least I'm kind of convinced that, maybe not all, but within the kind of within a reasonable approximation, we actually do pick up the main effects, which do have to happen. We are very good with mimetic systems now, can we do something more? And now let me give you a biological example. And this is the regulation of the activation of NK cells. Now, to do this, I would have to give you a little bit of biology. Now, NK cells are natural killer cells. They're part of your innate immune system. And what they do is basically they are searching for, they have this NKG2D receptors, which when they're activated, they actually find tumor cells, because the ligands on the tumor cells for this NKG2D are actually also upregulated, and they find them, while on normal cells, they're not. It's not that they don't exist. They do exist also on the normal cells, but at much, much, much lower concentrations. Okay, so now this is really me, a theoretical physicist, giving an introduction on biology, so please, all of the caveats to possible misunderstandings. But at least this is how I got the story. Right, now, so basically, when the NK cell is presented with the tumor cell, it actually somehow learns about the existence, probably through the binding, and this introduces a cascade, and this gets activated, fluctuations increase, the number of receptors increase, and so on, and so on, and so on. All right? Now, this process has to be regulated, and this cell is regulated by having another protein, which is called a KIR protein, or KIR family of proteins, and this KIR protein is actually down-regulating the whole thing. So you have a NKG2D and a KIR protein, each finding different ligands, and basically you have to integrate their signals, and the integration in this particular case, for this particular pair of inhibitory and activating signal, is believe that it goes really through the number of bonds, that it's a fairly simple integration process. All right, so the stronger the signal, the stronger the activation. That's the idea. Now, there is also another important thing, that the signaling pathway of the KIR and the signaling pathway of NKG2D are not actually talking to each other. They are somewhat separate. There are other activating receptors in NK cells, which actually have a signaling pathways which overlap. But that's a more complicated situation, and not the situation which I want to look at. I want to look at this situation where I have two proteins, because I'm actually interested to prove that there is a physical force helping or regulating this activation. So I don't want biochemical signals to interfere to each other. So we choose a system where the two signaling pathways are disjoint. All right? No interference. On the other hand, there is another measurement, which says that when you look at inflorescence, the KIR and NKG2D colocalize. Why? Why would they colocalize? Why would they want to have proximity? Is this proximity important? No. When they don't talk to each other. The signaling pathways don't talk to each other. Why do I need this proximity? So that's pretty much the question. I'm sorry for this really nasty picture. This is not really nice. But the question was, okay, why do I need proximity? And then we said, well, maybe this has something with this, generating the number of bonds and generating this signal, right? So maybe these membrane-mediated forces can be utilized for that effect. Now, the experiment was done in Mark's lab, and I will show you the experimental data. I mean, they're absolutely gorgeous. I think this is really, really lovely lovely case of mixing up mimetic and real systems. Now let's see what the theory is predicting. The theory is fairly robust. Here we have much more unknown parameters, so we have to guess a lot. All right? So I don't say that these are the exactly correct parameters, but I will tell you that there is a very robust result which tells you that there is if I put a bond and look at how likely is to make, or what's the affinity to make a short bond close to a long bond, and here I have to mention that the Keir and NKG2D construct are deliberately made at different heights. So this is at least a parameter which we know. So then you can see that there is kind of an optimal distance for the affinity of the short bond close to the long bond, and vice versa. You see this because it comes actually from the on rate. So which means that there is an optimal place. So I don't want to be too close, because if I'm a short bond and I'm trying to squeeze this long construct underneath, I can't do it. So I don't want to be too close. But if I'm too far, I don't benefit from these forces. So there is some kind of an optimum. Yeah? It's not actually too difficult to understand this optimum. It just, happens through the biomechanics of the proteins. And then you can actually calculate how many NKG2D and how many Keir protein bonds are actually being formed as a function between, of the gap between them. And you see that, of course, within this regime, you will be increasing the number of bonds actually if you are separating them apart rather than bringing them together. Yeah? Good. All right, this is what the theory suggests. Now, this is what Mark does. Gorgeous, really gorgeous. And I think everybody who does experiments, but also all theoreticians should really appreciate that. So Mark can take two dots, gold dots and one NKG2D. All right? So we have a super nicely patterned surface with very well control of the proximity. That's the parameter which we want to show. All right. Now, he can't measure this in microscopy. I mean, he needs super resolution to measure this. Mark doesn't have it. So I can't, unfortunately, this is now step number 2. This is what your work in progress. But what he can measure the expression of various activating signals, right? So he can measure what the cell is sending out, right? So he does, he checks for CD107A expression. Don't ask me, okay? I don't care, right? I mean, he basically tells me, this is a signal of activation, and you see, as we're increasing the separation between the dots, the activation is suppressed. All right, so you get really a signal from the fact that you are separating the purple and the gold dot, and the activation drops because the inhibition has actually helped in. And now he has done it on more than one activation. There is something which is called NF kappa BN, NF minus kappa BC. No idea again. Apparently this is upstream, this is downstream. Whatever this means. And then the interferon gamma, this one I can say no. Okay, I can live with that. But it's also exactly the same. Whatever you look, it is the same signal, right? So what in optical direct measurement, we can say it. From the response of the cell we do, there is a correlation between the separation of the proteins, and actually proximity is here not important, but there is an optimal separation in which the activation the inhibition of activation happens in the best possible way. Why? There are two effects which are actually happening. I am making more activating bonds, but I'm also, so as I'm increasing the separation, what happens is that it's not only that I am increasing the number of inhibitory bonds, I'm also increasing the number of activating bonds, because once you make an inhibitory bond, that helps the activating bond to be made. It goes two ways. So you're increasing the number of bonds, but you're increasing it such that still the inhibitory signal wins a little bit more. So That's all what happens. So as you come very close, there is this steric effect. They don't like each other. They can't be too much. So there are no activating, no inhibitory bonds. They're both suppressed. And as you're going out, there is more bonds, but more inhibitory bonds relative to the activating bonds. All right? So the signal is stronger overall. But it is plus minus. N times blah, activating, minus, and activating minus inhibitory. So it doesn't matter whether the relative, it's really what is the relative thing, okay? So what we think, it's really that the both effects are really taking place. But now we have another thing, right? And I have another 10 minutes, which is lovely, because I can then challenge a little bit more. So I can show you that, as I showed you here, I hope, because we've chosen a particular pair where there is no overlapping of signaling pathways, we play with one parameter and one parameter only, and we show these effects. Then we say, well, fine, yeah, these membrane-mediated things may be actually playing an important role. Now, this is very satisfactory, because when you're a theoretician, you find something where this thing actually works. That is nice. But then you are still a theoretician, and then you go and look at it properly, right? So then you go and say, well, actually I have introduced another parameter. I have a difference in length and difference in elasticities of receptors, which interplay with the membrane. What does this do? What would be the prediction of the model? All right? So now this is the system of interest. We have a long bonds, we have a short bonds, we have a membrane. And actually, when you look into the literature, there is a whole pile of biology which is happening with short, strong bonds and long, weak bonds. A whole lot. So if the cell if the macrophage is rolling through your blood, it actually attaches first for weak selectin bonds, and then it attaches with strong integrin bonds. Selectin is large, integrin is smaller. This happens for a number of pairs, in the T cell response, in the neural cells, all over the place. So why? So what is there, right? What is the physics tell you? What physics is behind that governs this idea? All right? Now, obviously, the nature has found in biology many ways to solve the same problem, right, of competition. And it uses this competition, always with different patterns because of different cells, but the principle should be there. So what is that principle? So you can look at the nucleation dynamics. This is the first thing which I thought could be interesting. If I have these intermittent long bonds, how long does it take to nucleate a strong cluster of short bonds? And you can see that, okay, this is the pure system of short bonds. Let's say, we are here, let's say 300 seconds. That's how long it takes me to nucleate for this particular set of parameters. I have no idea whether we try to use some reasonable parameters over here, but we get 300 seconds. This is not system specific, right? Now, this is the pure system, and this is when, so if I have a pure system, so if I have a pure system, it takes me something like 1,000 seconds. If I put a small concentration of long receptors, which are intermittent, and solve the master equation again, the same way I have solved it before for a single system, then I see an acceleration for the order of magnitude. All right? So now this is where you say the long bond is catalyzing the formation of short bonds. It's helping. It makes it 10 times faster. All right? Does it always work? No, it doesn't always work. You can create, if I make the long bond a little bit too stiff, right? What's going to happen, it's actually going to impede the formation of the short bond. So it actually produces an anti-catalytic effect. All right? And we being physicists, we can always grow a phase diagram. This is what we like. And then you will see that you play with two parameters. You have a difference in length of the long and a short, and the difference in stiffness of the long and a short. And then there will be a range where you have first stable nucleation and unstable nucleation, which comes from membrane parameters. And then you have basically positive cooperativity in this part of the phase space and negative cooperativity or anti-cooperativity in this part of the phase space. All right? So now, which means that if the difference in length is too large or the stiffness, so if the difference in length is too large, but you see this line is also going to go, so at some point you will not be able to nucleate at all. Okay? So, and you will also see this in the growth patterns, right? If I have a pure SNS, I will never grow. If I add a little bit of long bonds and look at the number of bonds which are being formed in a system, on the long-time dynamics, you can actually see the same effects. All right? That's nice. That's interesting. You know, one would actually have to look at the full patterns. So again, you know, you go and map the entire phase space as a pair of parameters, and you can see that as you are changing the length, this is the length of the one and the length of the second one, the red protein and the blue protein, and only bonds are formed, only bonds are short. Then you can see that by changing the length and keeping everything the same, otherwise you can really change the types of patterns which occur. And I would say that these are actually stable patterns. Right? These are not patterns which are metastable. This is something where we work very, very hard to actually find the lowest energy states. Now, and you can see that with variations of this, you can do this, but you can also do this with the variations of stiffness or with the variations of energy when you can get these ring-like structures and inverted ring-like structures and so on and so on and so on. So the patterning is very, very sensitive to the biomechanics of proteins, this being both the length and the stiffness, and not only the length. Because, have a look. Now, if I look at this part of phase space, right? I'm only changing the length. These proteins have the same stiffness. I can have differences in length of up to 10 nanometers, right? Over here, right? This is 20, 30. And I don't actually have, so the diagonal is when they're the same. This is 5 nanometers, 10 nanometer separations. I don't have macroscopic phase separation, even though I have a really large difference in size. 10 nanometers is a lot, right? On the protein scale. That's a lot of protein. I don't have it because they are the same elasticity. All right, can this be true? Well, sounds a bit odd, right? All right. So I asked Kea, I'll skip this because we are a little bit short on time. And I asked Kea, you know, is this possible? And then, but Kea said, well, you know, I can test it. So what she did is she took a vesicle and she made artificial receptors. She used DNA. She just used the DNA origami and used systems which have the same stiffness, but just different lengths. Right? So she creates receptors which are really the same. The only thing which is different is the length. And she makes them about 10 nanometer different in height. Now, if she allows only the short one to adhere the mean height of the membrane, you don't see the bonds, you see only the membrane, you can measure the membrane. So this is the separation between the two, the bilayer and the bilayer and the vesicle membrane is 10 nanometers. If you have only the long bonds, it's about 18 nanometer. All right? So pretty good, right? I mean, I have a good 9 nanometer difference in length in the pure phases. And now she starts mixing them, right? And now you see here for 8550 and for 8020, and you can do it for many more, but it doesn't really matter. What you see is you don't actually see any particular phase separation. These domains and height of a membrane gradually changes from 9, 10, 13, 17. It just gradually changes, but no obvious patterning within the size. Now we did, for this exact set of parameters, stiffness of the DNA is known. The length of this DNA contrast is also roughly known. We pretty much take this as a parameters for the modeling and repeat the simulations. And now we overlay the height of a membrane in the simulations and in the experiments. So the purple dots over here, these are the experiments. These ones are the simulations, all right? And now we do what this experiment can't do. right? We can actually look at the number of long linkers versus the number of short linkers, how many they are actually formed, right? And we can look at what is their actual environment. Because obviously, when I have a lot of long linkers, the probability to find the long linkers in my vicinity is super high, just by statistics. That doesn't mean that I have correlations. Or I don't have additional correlations. It's just by randomly putting things, you will have domains. But do you have really domains because there is a short bond somewhere there? That we cannot basically see. None whatsoever. So on the length scales of, let's say, 1, 2 proteins, we do not see anything which is given by presence of the long bonds. We have chosen this parameter space, so we knew that we should be getting what we got from the first simulations, but in a sense, this is also now what we see in the experiment. All right, with this I will close. I have another 3 minutes, and this is just enough to somehow try to convince you that you can, it is possible to do careful theory and experiments and bring them together in a quantitative way. You can do this, it's not easy, it takes a long time, as I said. I mean, this whole business probably took something like 10 years and maybe 10 PhD students and postdocs and so on and so on and so on. This is a lot of effort, right? But you know, everybody contributes a bit, everybody contributes a step, and this is how, you know, on a longer period of time, you can actually, you know, what I think is really, today, I think we have a fairly good understanding of this interplay of the biomechanics on the membrane and the protein level. Now, of course, there are many, many, many open questions. How this activity comes into place. Can I deal with the cross correlations? Because at the moment, I don't know how to write a minimal model for the active membrane, right? Because I don't have the cross correlations. I have no idea how to put it up. I don't want to go and write a minimal model for the active process because then I have things. Are these pumps? Maybe. Endocytosis, exocytosis? Maybe. Actin, polymerization, depolymerization? Surely. All of it contributes. I will never have a microscopic model for that. So I have to come to a way to deal with this on a slightly coarse-grained level. But I don't want to go so coarse-grained and say it doesn't exist, right? And that's the place, that's the next step. This is what we need to do. And for you, because I know this is a very long lecture, there was really, really many things. If there is something to take home, is that there is a strong interplay between the protein mechanics and the membrane mechanics, and there are strong membrane-mediated forces in these systems, and if you can memorize that, then I have actually achieved something today. Okay? Thanks.
All right. Questions? Go ahead.
Yeah. So in the many body problem, before this two binder types, before that you were using the binders of varying lengths and stiffness, right? Or was it just
Over here,
yeah, before this?
before this, up, over here. Here?
Yeah.
So here, no, this is really parameterized for cadherin. So we choose the size of the cadherin, we have a length of the cadherin on the extracellular side and on the intracellular, on both membranes. We have both constructs which are floating around have exactly the same length. And they interact with the fixed binding, with the binding affinity, which is measured in solution, and everything else is left to the model.
Okay. Also, so like, this is like in the real system, there will not be a time period where there will be no bonds between the receptors and membrane. So when you presented your model of Langevin and Monte Carlo simulations where you showed the curves were overlapping, so since they are of course Markovian processes, so I mean this is just a doubt. I mean one can start with the different bonds also, and they will just propagate the same, right?
This is your starting configuration. You can start with, certain amount of bonds. You can start with certain amount of domains.
Yeah.
You can start anyhow you like. This is, you read in your starting configuration, and you move on from there. Right? This is really how it works. This is the beauty of theory. You can do it. This type of initial conditions are so easy to
they won't matter.
So, but even if you start with, I mean, this argument has been around for a long time, that receptors, that the membranes are never completely free. And that has been used, in a sense, to keep them together, because if you leave them and you don't put the potential to hold them together, then they just separate. All right? Now, and earlier works, which also look at this long and short dynamics, actually did not have the fact that if I have no bonds, the membranes will stay at some finite separation. All right? So the way they kept it is by having already formed ligands and receptors to start with. All right? What you lose when you do that is you effectively lose a lot in the correlations, because it is really this restoring force of the membrane which provides quite a bit in these potentials. All right?
Any other question?
And that's why also in that systems you will not actually see the formation of the small domains, but the system is completely quiescent and they completely phase separate, right? Because there is nothing locally to prevent and actually create the line tension which stabilizes the small domains. That's the physical difference between this, whether I start, and it is not about whether with what I started, it is really an ingredient in the model. Okay?
Any other question? Okay, if not, then let me again thank Professor Ana for this set of 3 lectures, which were very useful for all of us. So thank you very much.
All right.